Welcome back. We're here to talk about chapter 15, which deals with specific immunity, also known as acquired immunity. Acquired literally because, you get sick with a thing, your immune system fights it off, and you have acquired immunity against that. All right. So lymphocytes, I should say lymphocytes start in the bone marrow. That's where all of them start out, whether you're B or T cell. That's where that hematopoietic stem cell precursor is going to be in the bone marrow. B cells fully mature in within the bone marrow, and then will migrate into the periphery and into the lymph nodes, while T cells will mature in the thymus, and then they'll move out into the periphery and the lymph nodes and station there and wait for activation. Once lymphocytes are activated, they will proliferate first, so it just means multiplying, and then they will differentiate into either memory, effector, or regulatory cells. And they're exactly what they sound like. Memory cells essentially give, your body memory of having encountered this antigen before. Remember, antigen is anything that can provoke an immune response. So anything that could provoke it. And that could sometimes, unfortunately, include self molecules, which would deal with autoimmunity. We'll talk about that in a little bit in the next chapter, but do keep that in mind. That is possible. Effector cells, those are the cells that are actually doing the job at the time, while regulatory cells are trying to keep everything in check so that we don't overreact to things. Overreacting of the immune system against things, that can lead to illnesses that something like toxic shock syndrome. That is essentially a super antigen, which is just an antigen that activates the immune system. That's what it sounds like. I don't know. Super antigen. Okay, so moving on, let me get my little pointer here so I can draw you guys some beautiful pictures. Okay, so any of the lymph nodes, any of the lymphocytes encounter antigen, they're going to first proliferate, and then after they've multiplied, those cells that have been multiplied are going to differentiate into those other things. So it's not like they differentiate like into one memory cell and it proliferates. No, the proliferation becomes first. So that's how that works. That's why I keep reiterating it. All right, clonal selection is removing anti-self. And then it also refers to proliferating the appropriate clone. So we're not going to activate and proliferate everybody every time anything bad is seen, right? So we're only going to, activate and proliferate the clone that has a reactivity against whatever antigen is being presented. So, and keep in mind, you can have activation of several clones. So there can be several kinds of antigens associated with one infection type. So that can happen as well. Again, antigens are anything that provokes a immune response, and they must be seen as foreign. The body sees them as foreign if they haven't been trained to see self as self, and then we eliminate the cells that recognize the self. If that doesn't happen, we'd be, trying to attack our own cells all the time. And so it's important that, we have distinguished foreign versus self throughout the development of these cells. All right, haptens are essentially molecules that are too small to act as proper antigens to stimulate the proliferation, the activation and proliferation of immune cells. They're too tiny. So they can be attached to something to make them bigger, essentially, so that we can, sort of like a carrier molecule. Then once it's bound to that, the body can react to that as a whole. And then, we've made antibody primarily, that can target that tinier thing. So whenever we do see the tinier thing by itself in the future, we've already got cells that can see it. We don't have to worry about activating it with that larger bit associated with it. Okay, that's how we would deal with something too small. That includes tetanus toxoid. Tetanus vaccine is a vaccine against a toxin. Tetanospasmin is the toxin. And so it has to be administered with other things. And that's why we have tetanus toxin, pertussis toxin, and diphtheria toxin all together. They're going to kind of all they're all haptens, and so they're all going to need help activating the immune system. So that'll all work together. Super antigens, again, they're going to induce overreaction by the immune system. We just mentioned that. APCs are antigen presenting cells. These are extremely important in the immune system, whether we're talking about the innate immunity from the last chapter, which is just like your basic, front lines fighters, or your acquired immunity. The antigen presenting cells are important for all of that. So macrophages, monocytes, dendritic cells, which are essentially monocytes that have developed into these tendril having cells that kind of grasp onto pathogens. And they live primarily in the skin area. And B cells themselves can also serve as antigen presenting cells. So MHC1 is a class of molecule that will present foreign molecule out, so foreign antigen essentially out. And this is, MHC1 is one that is found on all kinds of cells. So what I'm trying to say here is any cell in your body gets infected with a virus, something that is going to multiply inside of the cell. And this can also include if you have a cancer or something like that. But if the problem is originating from inside the cell, all cells have the ability to take the antigens associated with that internal intracellular infection and then pop that out on MHC class 1 on their surface. And then that can go interact with the appropriate cells to start activation of acquired immune response. This would be interacting primarily with CD4 helper T cells, the Th1 response. So Th1 is associated with MHC1. You can see that in the T cell section right below. So kind of looking at both of these here. So both of these things. Really good arrow drawing it in. Next, so that's on all cells. Endogenous antigen. So that doesn't just mean yourself. It means anything that could be growing inside of you, like a virus, for example, or cancer when things are going wrong in there. Okay, so that's MHC1. Any cell can do that. And that will essentially start the pathway to activation to kill things that are inside the cell. That is the job of the CD8 cytotoxic T cell. These guys are going to go out and recognize those like MHC1 presenting that antigen. They got triggered, told they can go activate by their friend, the CD4 Th1 response. So these are the ones that activate the cytotoxic T cells. They're all reacting to the same antigen. So these guys go out and find cells that have that MHC1 with that antigen presented on it. So the cell has this little MHC1, and then this is our antigen on the surface. And then the T cell comes along. This is the cytotoxic T cell. And it interacts with that, and it gets other signals from other molecules that are on the cells as well to confirm that it's doing the right thing. And then it releases these things called perforins and granzymes. Perforins perforate the cell. They're going to poke holes into the actual outside of that target cell. So our own cells, the ones that are infected with the virus or whatever, the ones that are pushing out that antigen on MHC1 is basically a signal to the cytotoxic T cells that, hey, something's wrong inside of me. So these cytotoxic T cells say, yeah, buddy, I know, I'm gonna have to put you down. So they come over and poke holes, and then the holes allow for the stuff inside to leak out. Obviously, that's bad for the cells, right? And then we have granzymes that just start breaking down the stuff that's inside of the cell. Once all of this happens, the cell knows that this is a losing battle and it will undergo apoptosis. Apoptosis is kind of a fancier word for saying that the cell is deciding to kill itself. So it's produces some proteins associated with breaking itself down, as well as just stopping, life processes. So that is the MHC1, the helper cell that's involved in helping activate that. That's the CD4 positive Th1. And then we have the actual effector cell that's going to be going out and interacting with this is going to be the CD8 cytotoxic T cell. So cytotoxic T cells have a marker called CD8 on them. These are literally called cell differentiating. That's what CD is. It's just a way for us to differentiate them, because otherwise T cell looks like T cell. So this CD4 marker is associated with the helper. And maybe this is stupid, but it probably is. I don't know. But to me, the number 4, because it has this and it has this shape to it, and then all you would need is this, that's like a helper. H is for helper. 8 does not look like a H. So I don't know. You could kind of look at it as maybe the 8 looks like 2 cells stuck together. Like, I don't know, that's silly, but whatever helps you remember it. So that's how I remember CD4 is the helper T cells. Now, essentially, we have an antigen that's going to be presented on MHC1 or MHC2, and a CD4 T helper cell associated with, Th1 goes with MHC1, Th2 goes with MHC2, which we'll get into in a second. Whatever it is, the antigen presenting cell, which could be the actual proper antigen presenting cells, the macrophages, the monocytes, the dendritic cells, and the B cells, those ones associate with MHC2. Any cell with MHC1, they can present on MHC1. Whatever it is, it's going to go out and interact with a T helper cell first, always a T helper cell first. And that T helper cell will go and activate whatever the next steps in the response are. So if a B cell is out there and it picks up an antigen that it recognizes with its immunoglobulin receptor, which is basically its antibody on its surface, but as a receptor, the B cell receptor. So it goes out and interacts with that antigen. And if that antigen also is triggering the appropriate CD4 Th2 cell, then we can activate that B cell. Otherwise, that B cell ain't going to be activated and it will not be doing anything. So T helper first, okay? T helper happens first, no matter where you're going. So we know we have our T helper going on first. We have our antigen as being presented. If it's being presented on MHC1, T helper has to interact first. So that T helper is going to be T helper Th1, because that's associated with MHC1. And that will activate the cytotoxic T cell response. Because we want to go out there and kill the cells that are infected. So it's our own cells we're killing because they're infected with things that's not salvageable then. All right, the next one, MHC2, because I know this is a lot, so that's why I keep repeating myself. I've found that repetition really works well, which is why I have the class set up the way that it is. So MHC2, those are only going to be presented by the appropriate antigen, classical, official, whatever you want to call it, antigen presenting cells. Macrophages and monocytes, dendritic cells, and the B cells. All right, just those guys. So they're going to be presenting stuff that was picked up from the outside by like phagocytosis or something like that. Okay, so they found it outside. Exogenous, that's what that term means. Exogenous means outside. Okay, so we have an antigen. It's being presented on MHC2 now by an antigen presenting cell. That antigen presenting cell will now interact with a T helper cell. Since it's MHC2 that is presenting the antigen, it's going to interact with T helper Th2 cells. All right, CD4 positive, they're always that. That's just all the helpers. The Th2 ones are going to be associated with antigen presented by MHC2. And that is antigen that we found outside the cell, exogenous. All right, what do they do after that? The T helper cells themselves, this is the case with the Th1 cells as well. Either one, Th1 or Th2 helper cells, everybody does this. All of the lymphocytes, T or B, once they're activated, they proliferate and then differentiate, right? So they're going to proliferate, and then they're going to go out and associate with appropriate B cells that also react to the antigen. Now, we may have already associated with some B cells as antigen presenting cells, but maybe we didn't. Maybe we just interacted with monocytes or macrophages or something. So now we have to go find the matching B cell that also reacts to the same exact antigen, and then we will activate them. They get activated. That clone that activates to that antigen will proliferate, and then it will differentiate into whatever kinds of cells. So B cells, same thing. They're going to have memory, effector, and regulatory cells. I don't talk much about the regulatory cells for the B cells, but they exist. The important ones with vaccines, obviously, are going to be memory. You activate some B cells, they proliferate, they differentiate. Some of those become memory cells right off the bat. So that whenever you see chickenpox virus again, or whatever, your body can just start producing antibody right away. You don't have to go through all this, activate the Th1 and activate the B cell and then proliferate and then differentiate. You don't have to deal with all that. We've already got our backup guys ready to go. They're already ready. They've already seen the enemy and know how to identify it and take it out. So that's our memory cells. And they are going to be able to become effector cells very quickly. And like I said, effector cells are just doing whatever the job is. It turns out that B cells, their job, their effector job, they become plasma cells. That's what they're called. That's what the cells are called when they are effector cells, the actual ones that are doing the job. Plasma cells secrete antibody. And you're probably like, cool, yes, we've heard of antibody, and we know that's what we need in order to fight off infection, but what is it and how does it work, right? So that's the best part of all of this. And keep in mind, with all of this immune response, we have regulatory cells just to keep everything in check so it doesn't go out of control and we don't start, if you start overreacting to things going on, like causing inflammation in an area, and we're starting to overreact, you can start to accidentally trigger reaction to self. And just because it's nearby and we've got all this inflammation, you're getting triggers to proceed, but it's not being kept in check. So that's why we have to keep everything regulated. That's what the regulatory cells are for. So back to our plasma cells, we know they produce antibody. Antibody is often drawn like this. We have our heavy chains. We have two heavy chains and two light chains. And that's just exactly what they are. The heavy chain is the long one. The light chains are the short one. And they're linked by disulfide bonds. They're covalent bonds. So they are really actually linked together by true chemical bonds. Okay, so that is what an antibody looks like. We have this part up at the top that includes the top of the heavy chain and the light chain. That's called the FAB, that whole top portion. That is a very variable region. So that part can change in all these different ways with all these different mutations that the body can put in itself, or by mixing and matching how the genes will meet up together and everything like that. If you saw in the video about it, which I highly recommend that you watch the video or at least read something about it. I think the video is a little bit better. They have it in the textbook as well. Please watch that video. It explains it better than I can, but it's basically gene reassortment in addition to inserting or removing certain nucleotides to give you a lot of variability. You can react to approximately, well, over 8 billion different antigens, which could potentially cover every single possible antigen in the universe, including self, of course. Many of these antigens you're not going to see, so those might be useless. So we kind of can lose our ability to respond to those as we get older. Okay, so that's the idea with how we have all these different antibodies against everything. Even though we only have 20,000 genes in the human body, we can react to all these different things. It's so cool to me. But basically, that's what makes each antibody unique. That FAB, okay, the variable region. The bottom part is called the FC. It's just the constant region. Okay, that's the C. And the AB, just think of that being like antibody. That's what makes the antibody respond to a specific antigen. The FAB does. The FC is just the constant region, and that's just going to determine what that antibody will be doing and what it can associate with. Okay, there's 5 different kinds of FC, constant regions, that make 5 different kinds of antibody. IgG, that's essentially the workhorse. That's the guy that we want to come fight for us. That usually comes on later in an initial infection. But once you see the thing again and you're reacting with your memory, it's going to be primarily IgG. These guys fight so fast. No kidding. People don't believe me when I tell them this. But if you have, okay, I had the chickenpox, right? I had the chickenpox when I was a little kid. And certainly, I'm sure out there somewhere in the world, I have encountered children that are having, are contagious with the chickenpox, whether they're having the symptoms actively or know it or not. But there may be some child, I could go up to a kid and lick his face and he has chickenpox and I wouldn't get any symptoms, right? Because I've had the chickenpox before. So we know I have antibodies against it. So my memory, what's going on there is my memory B cells are going to start multiplying up and start cranking out IgG, our workhorse. That IgG works a lot faster and is more effective essentially at fighting off immediate infection, kind of all over the body. But essentially, we'll just crank that out right off the bat and very quickly, so quickly that I get infected when I lick that kid's face and I never knew it. And you never see it. You've probably met people who have the measles and you didn't even know because your body reacted so quickly to it, you didn't even have any symptoms. That's how fast the IgG response can be whenever we associate with memory. Of course, it's not always perfect or ideal, but that is what the goal is with every single vaccine. So I think that that's pretty cool that you probably encountered things like tetanus or something like that, that you got vaccinated for, or maybe things that you've seen before, that you were exposed to before, and you see it again later and you don't even know it. So that is so cool. Anyway, so you're already fighting it off before you even have a chance to, have an inflammatory response or anything. Like your body's just that fast. Okay. I say that it comes on later in the infection, right? Because you get sick with something the first time you see it. That's the whole point of vaccines, is to put it in a controlled situation so you're not getting sick with it. So what you make first is an IgG. The first thing that you make is IgM. Igm Exists as a pentamer, and that just means this antibody that I had drawn for you over here. Let me just draw it out again, just so we can see it clearly. This is an antibody, of course. These guys will link to one another at the bottom and form a pentamer. I'm not going to draw the whole pentamer. You get the idea. There's 5 of those guys around one another, like in a star pattern. That's IgM. Now, IgM gives a little bit of a broad response, and it is easier for us to start producing right at first. It can be a little bit less specific. We can get very specific with our IgG and crank it out because it just exists as one antibody, like a monomer. Okay, it's just one of those Y shapes. IgA is a dimer. It is associated with the mucous membrane, so just 2 of these Ys. IgE is associated with allergies, and IgD is the B cell receptor. So that's actually on the surface of the B cells. So that's what all of those different kinds of antibodies are. Now, what do antibodies do? They are involved in, okay, let's start actually with, we'll start at the back. Let's start with antitoxin. Antitoxin. Toxins are just, maybe a small protein molecule, a peptide. They can be pretty small, like the tetanus toxoid, right? It's small. So antibody can bind with the toxin, and the toxin could be what was making you sick. That's the case with tetanus. The toxin is what we're worried about. So we have antibodies against just the toxin. They bind on the toxin. The toxin can't act as a toxin and do its toxin thing anymore because the antibody's bound onto it. That is an antitoxin. Anytime you hear about an antitoxin, it's antibody bound that way to keep it from functioning. All right. Complement pathway. We learned about the complement pathway in the last chapter. The classical complement pathway is triggered by antibody. So that's just referring back to that. Agglutination. Essentially, you can have antibodies that will interact with like a bacteria. So let's say that this is a bacteria. and it's covered in these antigens. So it could react with several antibodies all at once. And then this one is over here, and then there's more antibodies, whatever, you know, this is rough approximation, whatever. Well, you can eventually have all of these bacteria and antibodies kind of meshed all together because of how the antibodies are associating with them. Now we've got the antibodies kind of stuck all together via, sorry, I guess what I'm trying to say, let me reword that. We have the bacteria, viruses, the pathogen, whatever it is, all bound up via the antibody that is binding with the antigen on that pathogen surface. So it's just sticking them all together, and now they can't infect cells or anything like that, okay? Coding. If we have a virus, let's say you have a virus and it's got spikes on its little outside, and its spike looks like a circle, and like the cell, this is the cell outside of the cell. Remember, viruses are very, very tiny. It normally would match up with this receptor, right? An antibody comes along and can associate onto there. So now the spike can't interact the way that it's supposed to. That is coding. So that would just be blocking like association of the molecules with one another. Opsonization. Opsonization, we are putting antibody on the surface of cells and making it easier for phagocytes, those eating cells, our antigen presenting cells like monocytes, macrophages, and dendritic cells, to come up and try to eat up the organism. We've already talked about how capsules make that difficult for phagocytes. Phagocytes can't phagocytose a bacterium that has a capsule on it. So what basically this will do, allow us to put antibodies on there. Now the phagocytes can attach to the antibody. They couldn't to the sticky sugary coating of the capsule, but they can to the antibody that's stuck outside of that. So now the sucker is screwed. So that's the idea with opsonization. Opsonization is when the antibody helps phagocytosis, okay? Neutralization, similar concept to coating, except we're just kind of covering up the whole surface of whatever that pathogen is, and it can't do infecting, it can't do whatever else it's trying to do. Okay, so that's just neutralizing its ability to be. Okay, so that's the function. I'm going to circle it just to make me feel good about I circled it. Okay, that's the idea with antibody and what it does and what it is. So now you know why we need antibody. It does all these things for our body to help fight off pathogens. So essentially, whenever you first see an antigen, remember an antigen is anything that provokes an immune response. Our primary response is the first time that you're seeing it. We have a sort of latent period, which is first we have the whole innate response. Then we have to get our antigen presenting cells out to our actual like lymph nodes and everything to activate the appropriate T cells, T helper cells. And then the T helper cells have to proliferate, and then they have to differentiate. And then they can start associating with activating B cells or CD8 cytotoxic T cells. A lot of infections involve both. It's just one is going to be more prominent than the other. It just depends on the infection. So it's not like you just have only one or the other, the one or the two. You know what I'm talking about, the type cytotoxic T cell response or the antibody response, because they're obviously very different. Anyways, but you can have both. And they will respond appropriately to whatever antigen is being presented in whatever way. So primary response, we've got that latent period of all of that, the activating, the proliferating, the differentiating. Then that T helper cell will go find the B cell, and it will be activated, and it will proliferate, and it will differentiate. And then we will start making out the IgM, because that's what we make first, but it's not as good as the IgG. So then we have to rely on IgG to start coming out, and that'll be towards the end of the infection, and so we can clear the infection. So that's your primary response. But now we have our effector memory, our memory cells, so that whenever we see the thing again, we just activate the memory cell, and they can start cranking out the IgG because they've already gone through all the differentiation and everything. So that's why that can be so fast that you don't even know that you're infected with something. So there's all sorts of different kinds of immunity that can be conferred. We have natural active. That example we have is responding to illness. You just get sick from something just naturally, and you get like the chickenpox is when I had the chickenpox anyways. I got that naturally and I fought it off and now I have immunity against it. Natural passive is like whenever you get your antibodies from your mom, from your breast milk. We have artificial active, that's vaccination. It's meant to mimic the infection response and create antibody, but it isn't a natural exposure. And then we have artificial passive, and that would be similar to the natural passive where you get antibodies from your mom. Artificial passive, you're getting antibodies from like a bag. So it's like gamma globulin, things like that, that could be infusions that are meant to have antibodies to attack cancer cells or antibodies to help protect your body against, well, basically inflammatory, its own inflammatory response in the case of like Humira or something like that. All right, so we have all these different kinds of vaccines that can exist. We have the live attenuated. This means that we have a version of live pathogen that has been altered in some way that it shouldn't cause illness to you, but it does elicit a proper response. Ideal would be the killed versions where, you know, you would have no incidence of recurrence because sometimes organisms can revert back to the wild type, the type that makes you sick when you have live attenuated versions. Subunit is a vaccine that's made with part of the pathogen, not the whole pathogen. Toxoid, again, that's just dealing with an antibody that deals with a vaccine against a toxin and not the organism itself that makes the toxin. And then conjugated. So that's trying to link up, essentially giving you a vaccine of one pathogen, and we can't elicit a very strong response to pathogen A with any vaccine that we developed. Weak response. However, if we pair it in the same vaccine as this other thing we get a strong response with, now it turns out we can elicit a strong response from A as well. So that's just pairing things together to help the response along. And then you guys already know about the anti-vax movement. And of course, there are some side effects to vaccines, but there's symptoms from illness. So that makes sense that there would be side effects from vaccines. The problem is that we're trying to elicit an immune response and activate the immune system to be active, right? And a lot of symptoms that you feel from being ill are from your immune response, not from the disease, not from the pathogen. It's our response to the pathogen. So of course, sometimes vaccines are going to cause problems or the inflammation associated with trying to trigger a response to the vaccine can cause a bad response to something else like penicillin or something like that. Before that, then remember, we also had a discussion in the bigger lecture about the MMR vaccine and how that doctor had published a paper about the MMR vaccine causing autism. And he actually had to withdraw his paper because he had lied. It was false data. And so that paper has been redacted. That's actually a false paper, but of course, society isn't going to listen to that. So you can be educated about it, at least for yourselves. So that's it for specific immunity. And I will see you guys next time for immune disorders, basically.